regulated by many different kinases and phosphatases at many different sites. So in other words, a particular protein may have several different sites that can be phosphorylated or dephosphorylated in different locations on the protein. Each one of those events is going to have some kind of effect on the protein. And likewise, that means that a given protein can be regulated by multiple kinases and multiple phosphatases, each with its own specific target and each with its own specific effect. Now, one other thing I like to mention is kinases and phosphatases themselves are regulated proteins. Many times they can be regulated by phosphorylation, or they can be regulated by other means. So we do not want a given kinase or phosphatase active all the time. We want it active only in certain time. And here again, one particular, it's a form of leukemia, not the one I have, but it's a form of leukemia that is actually treatable long term because that form is due to overactivation of a particular kinase, and there is a drug out on the market that specifically binds and inhibits that kinase. If you take one pill a day, if you got that form, you take one pill a day, that can keep you going for years, possibly indefinitely. We don't have a long enough track record, but in many cases it keeps it in check. What was previously a fatal disease can be kept in check just by taking one particular pill every day for the rest of your life. And hopefully, and some people may live a perfectly normal lifespan from that. There's a couple other similar ones out there too in case the first one doesn't work. So anyway, you know, that's an example here. So kinases and phosphatases are very important, and they are regulated. They are regulated proteins too. So those are major points about phosphorylation. And once again, the vast majority of the proteins in our cells are regulated by phosphorylation. Now, the last version of covalent modification we're going to look at is something called acetylation. Acetylation is the addition of a negatively charged acetyl group, covalent addition. Acetyl group is acetic acid covalently attached to something. Acetylation is performed by a set of enzymes called acetyltransferases. What we have here to start out with is something called acetyl. Oops. Acetyl. Okay, got it. Acetyl coenzyme A. Okay, acetyl coenzyme A is this molecule called coenzyme A. We're going to run into this because it's actually extremely important in energy-producing metabolism. Basically, anything you eat for energy ends up in the form of acetyl-CoA. But we're going to look at it, it's got other roles. So you start out with this acetyl coenzyme A. It contains this acetyl group, this 2-carbon organic acid. And then what happens is the enzyme that does this tears the acetyl group off and transfers it to a protein. So now we have acetyl-CoA and a protein over here. And the enzymes that do this are called acetyltransferases. Because that's what they do. They transfer acetyl groups from coenzyme A to target proteins. So now we have the protein, and I'll just say AC, that's a common chemical convention for acetyl group. We've covalently attached the acetyl group to the protein, and then we have what's remaining is the coenzyme A, or CoA for short, without the acetyl group. Now, once again, this is an endergonic reaction. The source of the acetyl group and the source of the energy is acetyl coenzyme A. When you hydrolyze the acetyl group off, that releases a great deal of energy. It's enough to covalently attach the acetyl group to the protein, leaving the CoA to go off by itself. So once again, energy needed because it's a condensation reaction and a source of the acetyl group. Now, if we want to do the reverse reaction, which is an exergonic hydrolysis, we need a second set of enzymes. Here's our protein with the acetyl group on it, and we call this deacetylation. And the enzymes that do that are called acetylases. They perform the hydrolysis reaction, leaving the protein by itself, and then the acetyl group is torn off, released in solution. That's acetic acid, vinegar. So that's the process of deacetylation. Now, what we mentioned about phosphorylation also applies to acetylation. In other words, there are specific acetyl transferases. Each has a specific protein, targets. Each will add the acetyl groups only to particular amino acids on their targets. Proteins can and are regulated in many cases by multiple acetylation. They have multiple sites that different acetyl transferations can attach acetyl groups to. And proteins can be regulated by acetylation and by phosphorylation, the same protein. Now, for the most part, phosphorylation is the most widely used version of covalent modification. Acetylation plays a somewhat lesser role, but where you find a lot of acetylation as well as phosphorylation occurring is in the nucleus. Okay, now you do get some acetylation in cytoplasm, but the nucleus is where acetylation really takes off. A lot of proteins in the nucleus are regulated in part by acetylation, especially proteins we call the chromatin proteins. Now, chromatin refers, or chromosomes when they're packed up like we saw in our onion root tip things. Chromatin is DNA with a whole slew of proteins associated with it that organize the DNA, help with processes like transcription and things like that. So DNA plus a whole bunch of proteins is called chromatin. The proteins that go in there are the chromatin proteins. And they're very diverse, but one of the most abundant kind are what are known as the histones. Histones are proteins that are involved. They are the fundamental structures that assemble the DNA and organize into chromosomes. DNA wraps around little balls of histone proteins, forming structures. If you took genetics, you may have heard of this. Structures called nucleosomes. I'll put that down here just for... And then the nucleosomes pack together in various ways to make what we call higher order chromatin structure. It packs, compactifies, and organizes the DNA into chromatin and chromosomes. Those are the major ones. There's others too. Now it turns out, not only do they organize DNA, they also bind so tightly that they block sites that would be that transcription factors could bind to. So they block transcription. Now, when a gene is about to be transcribed, in other words, you want to activate that gene so it makes messenger RNA or whatever kind of RNA, before you even do that, the first thing you have to do is do something with those histones so you can push those nucleosomes off the critical sites that these transcription factors bind. Okay, how do you do that? Well, histones have lots of positive charges, so they grab onto DNA and hold really, really tightly. DNA is negatively charged. So what you have to do in order to push these nucleosomes away from critical sites so transcription can take place. You phosphorylate and acetylate these histones at multiple sites. So what are you doing? You have a positively charged protein. Now you're sticking a whole slew of negative charges, canceling out the charge of the protein. Now the histones don't stick to the DNA as tightly. And now other enzymes, they can go up there and push those nucleosomes down the DNA, releasing and exposing the sites that the gene-regulating transcription factors can bind. If you don't do that, the gene will not get transcribed. So in this case, these very important proteins, they have to be regulated both by phosphorylation and by acetylation. Stick as many negative charges on them as you possibly can, and then you're good to go. So acetylation is another important version of covalent modification. Okay, one last thing with protein regulation.